Well, good afternoon, everyone. I'm Lisa Benkin. I'm a regional extension educator in crops out of southern Minnesota. And I want to thank you again for joining us to the fourth in our series of the essential row crop management webinars that we have been putting on the last couple of weeks. Today, as our fourth in series, we will be talking about the soybean gall midge, a very perplexing and difficult new pests that we are facing here across the region. Today, our special guest is Justin McMechan from University of Nebraska, as an extension entomologist based out of Lincoln. I've got a few housekeeping details, so I know he's bringing up his, we'll let Justin bring up his PowerPoint presentation, but a few things to remind you again about the webinars. First of all, thanking the effort. We've got a collaborative effort with the University of Minnesota Extension along with Iowa State University. Our coworkers across the border, we've got Angie Reeks-Hintz and Megan Anderson, Liz Stahl from Minnesota, and Phyllis Bongard from University of Minnesota. Again, a cooperative effort, been really fun to work with and put this whole series together again. We also have sponsors for our webinars, and today's sponsors are the Iowa Soybean Association and the Minnesota Soybean Research and Promotion Council. So we do thank them for that support. Throughout the program, we will put information in the chat box in regards to self-reporting our CCAs. We've got links to the SciBox for all of the resources that we have and other concerns that you might have. If you have like a technical issue, please put that in the chat box. At the end of the seminar, we do have a question and answer time, and there is a box too that we would appreciate that you put your questions, write those in that question and answer box, and then we will get to those at the end of the webinar. So with that, I'd like to get us started. Again, we have Justin McMechan, extension entomologist with the University of Nebraska, Lincoln, and again, focusing on soybean gall midge. So with that, Justin.
Thanks. And I appreciate the invite today and the opportunity to talk about soybean gall midge. I hope everyone can see my slides. Please let me know if you can't. And so I am one of a number of people that are working on this right now. And so Aaron Hodgins is on here and will help me out with some questions. I'm sure we're going to receive some difficult ones today. Adam Varenhorst in South Dakota, Bruce Potter in Minnesota, Tom Hunt and Bob Wright here in Nebraska, and Bob's on as well. And so this insect's new to all of us since its discovery in 2018, and it's been a battle to work with this thing, but we're learning and gaining a lot of ground. So kind of today's goal is to provide everybody with kind of an update on where we sit with this insect, what we're learning this season. You're going to see some photos taken as old as 2 or 3 hours ago, but provide you a basic overview of what we've been seeing. So for those of you that are unfamiliar with this, or maybe this is the first time you're hearing about soybean gall midge, we think this is the same thing that showed up in 2011 in some isolated fields in northeast Nebraska. Tom Hunt had found it up there. These fields had received injury or were diseased, and we considered it a secondary pest of soybean. And so there was generally a low level of concern all the way through 2016. Obviously documented in South Dakota, some associations with potential diseases there, and then in Iowa and east central Nebraska when I started in 2016, but none of us were raising a large alarm with this insect during that time period. We found out later some growers were kind of dealing with this, but on a very local scale. 2018, as many of us know, this thing showed up much earlier in the season. So prior to that, it had been a late season pest, mostly noticed in August. In 2018, we were getting calls in late June and early July. It was widespread. We were seeing injury in a lot of fields like the photo you see there, and we'll get a close-up of that later. But widespread infestation, significant injury. And this led to us pursuing the identification of the species. So prior to that, we just had known them as orange larvae, likely in the genus Rosiliella. But with Ray Gagne and scientists from Japan, Junichi Yukawa, we were able to identify this with the capture of adults here in Nebraska, leading the identification of a new species. So this should set the tone for all of us that this is really new and there's a lot to learn, a lot of gaps in knowledge that we need for management, but a lot I think has been gained in the last year or two. 2019 season, we picked up 34 new counties. Those are the light orange counties. The dark red are where we noted it was located in 2018. And this was from a help of probably many of you that called in, sent photos, said that, it's in this particular county. We've already added one in 2020. And so Lauren Botine, working with Corteva or Pioneer, helped us with that one. I'll be making a trek through the Nebraska counties here pretty soon to see if we can find this effort and the efforts here going forward. A large part of the funding that we're utilizing is through the North Central Soybean Research Program and United Soybean Board. So those two groups came together and have really come through helping us to gain what is a lot of much needed information. So we're attempting to survey all 12 states that were under the region, at least when the grant was formed, and identify if it's present in any of those states. Some host plant resistance work, which is the U.S. germplasm, over 750 lines. That's currently in Iowa. Oh, I see Iowa's name is missing from the thing. Iowa State's there. You all know where you are. As well as Nebraska and Northeast Nebraska. We have that germplasm planted, and then we're doing this adult monitoring, which will be a focus of the tail end of this presentation, to help us with the initial management practices on this particular insect. I know there are a number of states that may be present on here, so I wanted to briefly bring up white mold gall midge. So Bob Koch, along with Bruce Potter and a number of others, Janet Knodel in North Dakota, worked to identify what is the sometimes misidentified species, Cartiomycolocola, or the white mold gall midge. We haven't seen it in Nebraska yet. I know I have some samples coming in from Iowa, which may or may not be this, but this is one that we may run into. It's associated with white mold. It's not a major economic pest like soybean gall midge has shown its potential in areas. And so this is just one that if you come across some orange larvae in soybeans, it doesn't necessarily mean it's soybean gall midge. Go ahead and contact one of us. We have molecular techniques here in Nebraska to determine or separate those two species. Part of the goal today was to really discuss some of the symptomology of this insect, and what you're seeing here and in the next couple slides is the more extreme measures of that, but it's easier, I think, to tell what we should be looking for. And so starting from left to right, you have a soybean stem which is darkened and concaved at the bottom. That is typical symptoms of soybean gall midge. Peeling that soybean stem and that outer tissue off reveals orange and white larvae if they are present. You can see the white larvae there in the third photo. That's the diagnostic feature for soybean gall midge. Out this time of the year, you may find injured plants that don't have any larvae in them. So if that's the case, keep looking until you locate those larvae. They cycle through these plants a few times during the season, and we've come up with a few that are injured but don't have any. And what they're doing is they're feeding initially on the outside of the plant, working their way into the inner, starting the phloem, the sugar portion, moving into the xylem or water conducting part of that plant, and essentially cutting off all the circulation to that plant or its ability to cool itself, and causing the plants to die, which is this last photo here, which is taken from this year. So just to give you some perspective, here in Nebraska, that's the Saunders County 2018 site where we got the initial phone calls of significant injury occurring earlier in the season. You see a lot of plant death. If you see something like this, sometimes the initial reaction, probably not this extreme, but a few dead plants, sometimes to a lot of people says, oh, it could be Phytophthora, which is certainly a possibility. And if those plants haven't been dead for too long, trying to pull them out of the ground is quite difficult. They're anchored in really well. If it's Phytophthora, they pull out fairly easily. So that's one way to distinguish them. But ultimately, getting to the live plants that are there, looking at those for larvae can help distinguish those two. 2019, this is actually right across the road from that original 2018 field. For Nebraska, pressure went down in 2019, and we are certainly up this year in pressure, seeing a lot for what is late July pressure. This is Cass County, Nebraska. You can see here. And so if I bump ahead one, I took these photos literally 2 hours ago, or 2.5 hours ago, I guess at this point. This is our Davie site in Lancaster County, Nebraska. The injury is hard actually to photograph for this because as you look at this top photo, the vegetation here tends to hide the dead plants as we move out into the field. But obviously we're getting a lot of plant death on the edge of the field. And that's evident looking down on the top here. So this is actually a planting date study that we put in with and without gaucho or imidacloprid seed treatment. And you can see here that these plants have suffered significant mortality. This extends for a couple miles in either direction on fields with varying levels of injury or loss. I want to briefly step back from what I've been discussing, which is all soybeans. It's our target crop. It's a cash crop we grow. But soybean gall midge has been found on 2 other additional hosts. I've called them alternative hosts, I think, in the past, but we don't know the relationship between what's found on soybean and what's found on sweet clover. And we have a master's student here at the University of Nebraska, Ravneet Kaur, who is following up on some work from Amy Haver, which Amy discovered these 2 hosts in 2019. We ran through the gamut of larval morphology, through the Ray Gagne, who's a taxonomist with USDA, retired now. We did DNA sequencing at the University of Nebraska here, and then reared some adults to confirm these were in fact the same species. But we don't know their ecological role to one another. We're not certain that sweet clover is a significant risk because we find it in a lot of ditches. It's quite an abundant host. It's a lot harder to find them on alfalfa. But we have found a few this year. The photos here, this is what I call a healthy, non-symptomatic sweet clover plant, turned upside down. I could invert the photo, but I think it's even weirder to look at the sky on the bottom. And then these 2 plants here were both infested. And so I'm not entirely sure at the moment if the blackening has anything to do with soybean gall midge, but these grayed out areas, off tan colored, that kind of merged line there, if you peel those away, we tend to find the larvae under those areas. So if you're walking around and see some sweet clover, even if you're in an area where we haven't found soybean gall midge on soybean yet, this is of high interest for us. And if you locate any, depending on which state you're in, certainly contact the people that are in your state. I have a website for reference for that. But we would love to get samples of these to determine or confirm that they are in fact soybean gall midge when we may see other things on these hosts. We don't know a lot about what's on them to begin with. For scouting, you can focus on corn-soybean interface areas next to dense vegetation. Anybody who's dealt with this, those densely vegetated area, whether it's small trees, brush, grass, tends to be the areas where we see initial injury and extensive injury show up. Early planted soybeans, I would say that in 2019 with a lot of confidence. For Nebraska in 2019, our extended emergence has pretty much taken every planting date all the way through to June 1. And so that's not the case for us, but focus on those early plantings. They're at higher risk for getting infested. And, we're right, and for Nebraska here, we're kind of sitting in the R3 stage now. Things are evident driving around that fields are infested. So this is a good time to look and see if they're there. And that's more for the next year. Management this year, we're well past that, I think, at this point, more focusing on what the next year looks like. So one of the cornerstones of the work that all of us have been doing has been on the Adult Monitoring Network. This is coming from a number of funding groups, as I mentioned earlier. And what we have is this network. It's approximately 37 sites where we track adult emergence using cages. They're actually corn rootworm emergence cages that seem to work well for soybean gall midge. They have little jars on the top, and there are a number of adults that you can see in this photo. There's a close-up of a female. They have an orange abdomen that's hiding underneath those wings. This is a service we provide to all of our clientele. If you're trying to, or dealing with soybean gall midge and trying to make management decisions, we can send out through both email, text message, and phone call an automated system on when soybean gall midge is emerging in a given area. And this is the overwintering emergence from last year's soybean fields. On top of that, we have a website, which I believe will be pasted in the chat area if you need it for later reference. The website, and hopefully all of you can see that, has information that we're requesting information on injury scores for Mitchell Helton, which he's on here as well. He's a master's student from Iowa State University, and he's looking for injury data taken preferably, at least once a month to maybe once every 2 weeks if possible, and trying to correlate that with injury and yield losses in the field. We have a scouting video. I think for the sake of time, I'll avoid it for now. Feel free to watch this. This shows you how to scout a field and what to look for. So that's good information. And there are a few other things in here. Our last updates, our current first generation map from this year's soybeans, where we have numbers here that show you the average number per cage. The numbers are quite high once we get into emergence from this year's soybean. And so I think it would scare a lot of you to see the same type of numbers, this is a total number per site. And so we post that average. We can increase these numbers because we know exactly where to put those cages. We have infested plants. For overwintering, we just have soil and the stubble of last year's soybeans. And so we're really guessing on a lot of this. We have a grant in that hopefully, if we're lucky, we'll get funded to try and increase our accuracy on those cages to detect first emergence. So that's a good resource if you're looking for information. All of our contacts are also in there too, listed on the bottom. And so if you're looking for somebody specific in your state, those are people who are currently working on this. Timelines, this is the timeline from 2019 when we detected adults on June 18th. We were about 9 days to finding white larvae, 12 days to the transition to orange, and 20 days to initial wilting. It is somewhat similar to this year. We're realizing there's a landscape difference on when that occurs. So that dense vegetation or a lot of trees can actually, depending on where you're looking in the field, they may show up a little sooner. And as a result, maybe adult emergence occurred sooner in some of those areas. But these are things we're still sorting out. One thing I wanted to highlight to you to address, kind of the plant-to-plant complexity of soybean gall midge is this time lapse. And so many of you may have seen this, but focus on these two plants right here. All these plants have some degree of infestation, but these two in particular, they're both infested at the time the video starts. It's early July. And you notice the one plant is starting to lose its petioles. The other one seems to be hanging on fairly well. And here by about mid to late July here, we have death of this plant. And so obviously the questions are raised. Why is one still alive and one dead? Could be nutrition. People have brought that up in the past. Could be larval number per plant, the status of that plant at the time of infestation. But this is the complexity of trying to deal with soybean gall midge. This is a basic life cycle. I've given a number of these talks, and everybody always asks for this, which I think is great. And I appreciate the emphasis on the ecology of this insect. I had originally eggs from a related species in the Poland, Denmark area. We believe these are eggs of soybean gall midge. We're going to run PCR on these to confirm, but a number of us have seen these this year. And so these eggs are laid into natural openings, fissures in the plant, or wounds from hail. I have soybeans in my backyard. My wife let me plant them, which was nice. But we got a hailstorm that came through, and all the points where hail contacted the plants, we have infestation of larvae in those areas. And so that's been a concern, that hail might increase the number that overwinters or infest these fields. And as a result, possible injury for the following year. Purely a hypothesis at this point. We're not sure how that all plays out. We have 3 instar stages that they go through. First and second are white. Third, and there's kind of a transition to orange. All of these feed on the plant. These are the easiest ones to find. I can tell you the number of times that counting, at least in the field, it's very hard to see these. Under a scope, you even have to tilt them sometimes to catch them on the top side. They eventually fall off and pupate in the soil. They form little silken cocoons. And then just last week, 2 of the students in my lab were able to identify the pupal development stages in soil samples. So you can see the transition here through to adult emergence. And this is a broad estimation, somewhere between 3 to 5 days, probably closing in more on 3 days of survival for adults. They don't feed. It looks like they just need water as they move between sources. And that's, again, a female midge. You're unlikely to see the midges. We use the cages to capture those. I've never been successful with nets. Some others have caught a few, but likely numbers are pretty high in those fields. So this is eastern Nebraska's sites, and all sites combined for the adult emergence. And this just shows you the broad pattern of emergence, which it kicked off on the 10th near Louisville, Nebraska, picked up fairly quickly, and then kind of did the zigzag back and forth. We varied who checked cages, but not in that type of pattern, so I'm not sure why they bounced up and down like that. We have some theories on this depression point here where things really dropped off to pretty low numbers. Possibly the heat and dry weather may have delayed some of the emergence, and then of course they picked up and finally finished here on the 15th, I believe is their last day of emergence. That is a pretty long emergence curve. I'll show you as an average across the sites what we were seeing. But unfortunately, before we were even done with overwintering in the adjacent fields, we started getting emergence from this year's soybeans. And now you see a new y-axis showing up here with first generation adults. This is total across the site. As I mentioned, the numbers are a lot higher. The cages represent where they are here. So last year's soybeans for overwintering, this year's soybeans for this generation. You can see kind of low levels here, and then they picked up pretty dramatically here. So first emergence here was on the 30th of June, which is earlier than normal. So we're obviously, a lot of us are here to discuss management and trying to determine how to manage an insect like this. And this is the slide that you should keep in the back of your mind as we look forward on soybean gall midge. And the key word in here is duration. So ideally, with a lot of insects, we like a short duration period, an opportunity to really, capitalize on the population, either with acute practices or utilize something like planting date to limit their ability to get in. Unfortunately, the length was quite long this year relative to last year. So last year, we were an average of about 16 days per site. That was all the way down to just 2 or 3 days and up to, I believe, 23 days. In 2020 here, at least in East Central Nebraska, we ran 25.6 days of average, and the range was anywhere from 19 to 34. Now, if you're sitting there and thinking, well, man, did they just have volunteer soybeans or something in their cornfields? We scouted, checked, and made sure there was no volunteer soybeans in these fields where we are continuing to see emergence. So this is, in fact, still emergence from last year's fields, and they didn't complete a cycle out in those fields. So this poses significant and considerable challenges going forward. And hopefully it was the dry weather that drove this longer duration, and that may not be the case for the future. This is new as of this past Sunday. We did a survey. My wife and I, she's the insect toxicologist, and so she's also got a population genetics background. And so her and I are piloting along with Ravneet Kaur, who I mentioned earlier, a master's student, to understand the population genetics of soybean gall midge. And we're getting help from Iowa and Minnesota and others to get samples so we can determine how much genetic variability there is. We went adjacent to soybeans and adjacent to corn fields, which would possibly represent for the corn last year's soybeans. And you could see here in our trek, we have, first of all, no white dots, which was particularly surprising to me. Regardless of the random stops we made, we maybe had no wilting or low presence of wilting or death along the edge of these fields, but we found soybean gall midge in every single field we stopped at. And that's really concerning going into 2021, that there's a broad population out there. And so that's a concern. Sweet clover, if you heard me talk last fall or winter, I said it carries a low number of adults usually. That was not the case. We have to actually still count this one in particular, but there's a very large number of adults on those plants. And looks like there's 2 stems. It's just the stems have a long area of infestation, so we cut them in half. On soybeans, we've been seeing anywhere between 1 and 118 adults per plant. So a summary of management. Perhaps many of you are sitting there going, man, I really want to, tell me what to do for 2021. I think we have to wait on that conversation to see final yields this year. We are seeing some promising things this year, but ultimately this continuous source of adults may suppress out a lot of that opportunity. So what we suggested is plant your high-risk fields last. There's an indication that seed treatments or in-furrow treatments may provide some level of protection early season. Foliar applications have shown some larval number differences, and so there's some potential there. None of those tactics alone last year, this year, is enough to reduce all the injury in the field. And then this was from 2019. Mowing field borders seemed to help. That grower has repeated that study, and we're hoping to collect some information from it. So that's what I had. I encourage questions. Aaron's here as well. We'll do our best to answer them. I'll leave a larvae flinging through the air to spark some of your questions. This was while we were trying to count. They like to fling themselves through the air. So questions, comments?
Well, thank you, Justin, very much. We really appreciate. There's a lot of information that you are sharing with us, and reminding the folks that are listening in today that put your questions in the question and answer box. We will get to those, and that there, of course, we have the resource with the SciBox putting in your presentation, along with additional resources that should be helpful, like your scouting video. I watched that earlier, so that could be very helpful as you're out in the field. There are a few questions here that I have that I'd like you to address, but thinking about the scouting, since you mentioned that with the video, people should be in their fields now scouting. Is this a good time? And where would they focus to start, just to get a feel for what might be in a field?
Yeah, so for the scouting, now's a great time to scout. We don't want to go too late in the season as things start to senesce and mature, but they have a good 3 weeks to a month, I think, for some scouting. This is probably one of the only good things about soybean gall midge is scouting is pretty easy. So take one step into that field, or in fact, even the first row of soybeans. That, at least for Nebraska, I believe for Iowa, Aaron can comment if that's different, but I think most of us are on the same page. That first row or second row is the place to focus on, and you want to go directly to the base of those plants, like we showed in that other video, or that earlier slides where we had the photos of all the different types, and you could just snap those plants. Sometimes pulling them up, they'll break off, but looking for those orange or white larvae at the base of those plants is key. High risk areas are obviously next to last year's soybean field, or what's usually this year's corn field. And I typically focus on areas where there's dense vegetation. One last caveat in that is that if you have an uneven stand or height of plants, and you're really looking to find them, go to the tallest or largest plants along the field border. That's, at least from our experience, the most likely ones to be infested. Our planting date study got hit pretty hard, and one of our questions was, are they going after these larger plants that were planted earlier? And so hopefully that provides some good advice on that. If there's questions on it, I'm happy to entertain them if there's follow-ups.
Another one, thinking about the fields that were identified with the pest a number of years ago. If you come forward and looking at, soybean, corn, soybean, what do those fields look like today compared to when they were first found? Are they much worse? Has there been anything done that has improved the population, or still finding the gall midge in those fields? So if you think of the first finds, what has happened with those fields over time?
Yeah, so we see a lot of ups and downs with soybean gall midge. So I could say that some sites were receiving pressure in 2018, had been noted with presence in 2016 and 17, and since 2018 they have consistently shown what I would say is significant pressure from soybean gall midge. Other sites, like I have a site out near Plattsmouth, Nebraska, which the grower planted soybeans back into it. So we are so grateful for growers willing to put their fields at risk. But this grower planted 2 years of soybeans on a high pressure field, and we were certain this would be a really good high pressure spot to understand treatment response. And that site is lower in pressure this year for whatever reason that might be. And so we do see some continue and maintain the level, others decrease. And we've seen also spots where, like north of Columbus area in Nebraska, where they simply detected in 2019 And then I got several reports this year in 2020 of a lot of pressure in those areas, probably even, to the extent or what I've been hearing, like the site I showed earlier. And so there are a lot of variables at play. Last year was planting date. A lot of our conversations in the fall about I had it, my neighbor didn't have it. A lot of that boiled down to the time of planting. So there's a lot at play, and it can go up or down on some things we know and some things we still don't understand.
I do have a couple of questions that are in regards to seed treatments. The first one are there any commonly used seed treatment insecticides that are effective or showing some management control, or common use rates or increased use rates? So anything right now that you're seeing with the seed treatments and managing this pest?
Yeah, I was certain that question was going to come up, and this is, everybody that's watching this should be really cautious about trying to interpret this information. But I have a slide to share. Since this is gaucho, so imidacloprid. This answers the front end of that question, but don't hold your hat on this till we get to the end of the season. We're looking at two side-by-side graphs here. One is the frequency of infested plants, the number of infested plants out of 10. We have treatments on the bottom here, planting dates from late April through June. And then we have with and without a gaucho seed treatment, which is at a grower labeled rate, so kind of a mid-level rate. And what you see here is the early planting dates, lots of bouncing around, both in terms of frequency of infested and number of larvae. So no real differences here. As we move to the 12th, and this is the Davie, Nebraska site that has pretty high pressure, we see a decrease both in infestation and in the number of larvae, and that follows through to the 22nd. And in June 1st, when this was taken at the end of June, we didn't have any presence here. So that looks great. But that photo you saw is this study with continued adult emergence for what was 26 days at that site, and an overlap with first generation. They have continued to pound these plots. And to be honest with you, I can hardly tell the difference with and without gaucho now, because adults quickly hit these plants as soon as they reached the V2, V3 stage and had any of that fissure development. But it's a hopeful early sign that this is the start of trying to understand this, and maybe our abnormal year with long emergence, maybe this would look better. The Nebraska Soybean Board checkoff dollars have funded this for the next 3 years, and so we will be digging into this pretty heavy to continue to understand this total response. But everybody should be cautious. Wait till we pull yields on this study and see if we were able to protect any of that yield under a high pressure situation.
Okay, the question that follows with that is, you've somewhat answered that, but it says, is there any hope of a long-term systemic insecticide for soybean plants?
Yeah, so Aaron and I and several of the others that are working on this are in contact with companies quite a bit over this. And this is one example. We have a Cruiser Max study too. The issue is at best, we're looking at about 30 days of protection with these seed treatments. And depending on time of planting, obviously, like this particular planting here ran in, right into the front edge of soybean gall midge. These had already worn off really by what I hear from companies in terms of seed treatment protection by the time they showed up. And so we just can't carry that long protection with such a long duration of emergence. And so I think, and like we've been saying last year too, there's no single tactic in front of us for soybean gall midge that's going to completely manage this insect. It's going to be a combination of tactics like the planting dates we see here. We don't want to hammer these fields continuously at the start of the season. Our conversation with our multi-state group on soybean gall midge, one of the big topics that came up was spider mites. And so continually hammering these fields is going to cause issues, even if it's just field borders. We'll need to come to something else for that. I believe I have this one other slide. This is just a little bit of understanding of white and orange differences in larvae. You can solely focus on this one. here, sorry, let me highlight the right one, that we were having difficulties with early infestation. Our gall midge was on gaucho treatments. White larvae are the younger larvae, their equivalent. I'd show this just purely so I could get to this, which is funded by North Central Soybean Research Program and United Soybean Board, which is a germplasm screen for 750 lines. This is part of our hope going forward, is that we can identify some resistant or tolerant lines to stack up maybe with even other things that might be necessary. Aaron and Mitchell are running this in Iowa. I have a site here. Tom Hunt has a site in the Northeast. And so we're really hoping to come away with something that can stand up to this insect and provide us some additional support that's not insecticide related. The mowing and other things help as well. So it's a long battle. We're not going to have any quick answers. That's pretty clear at this point.
One other quick question on that. It's in regards to yield. What kind of yield losses have you seen in some of these fields? Grant, it may be only a portion of the field, but have you seen complete loss in some areas and then less as you move out, or how big of an area in a field have you seen?
Yeah, so if we talk about plant lethality, which is obviously large losses when we have plants dying in the field, that's typically confined to the first 50 to 100 feet under high pressure fields. So these are our worst fields. One of the things that concerns us, and without a really effective means of controlling soybean gall midge, is those plants that survive and the level of injury that occurs at the base of the stem. What is the sublethal loss of those plants in terms of yield? We don't have a good handle on that, and there are no check plots out there to say, oh, this is what we would have had for yield. Growers in 2019, which would be the 2018 season's yields, told us anywhere between 17 and 31% losses. Those are pretty extreme losses. These were growers that obviously had a lot of issues, but that was what they were seeing on their impacted fields. We need to get a handle on this, I think, a better handle on it. It's just been a difficult thing to do when we don't have any way of measuring against something that is not impacted. Aaron, if you have something to add, please feel free to add on that.
You're doing great, Justin.
Okay.
Yeah, so sometimes, depending on the size and the shape of the field, there's really noticeable yield differences because the field is small and not a square or a rectangle. But the bigger the field is, it seems like it does dissipate, according to yield monitors. So it kind of tempers as it goes if you have a larger field.
Well, thank you very much for your time today, Justin, and for your input, Aaron. We really appreciate all of our listeners in taking in the program. Please, a reminder again that we have this information that will be posted in the resource SciBox. Feel free to look at that. The videos, as far as all these are recorded, that should be up in just a few days. So then you can watch this again or look back at the ones or share it with other people so they can take in the presentations. You will also receive a survey very soon here, again in a day or so. We'd appreciate your feedback to see what you think of our essential row crop management webinars and if these have been helpful and what you've maybe learned from these webinars. So thank you very much to everyone for participating and enjoy the rest of your day.